Hello, everyone, and welcome to today's webinar titled Boosting Signal to Noise in Single-Cell RNA-Seq Through Molecular Depletion. This webinar is a part of the 10th Annual Genetics Virtual Week. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Jumpcode Genomics. For more information about Jumpcode Genomics, go to jumpcodegenomics.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dante DeAscanis, Research Associate 2, Research and Development, Jumpcode Genomics. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Dante, you may now begin your presentation.
Hi everyone, my name is Dante DeAscanis, and I'm a Research Associate 2 here at Jumpcode Genomics. And today I'm going to be talking with everyone about how we can improve the signal-to-noise ratio in single-cell RNA-seq data using one of our products called CRISPR-Clean. The official name of our product is called the CRISPR-Clean Single-Cell RNA Boost Kit. For those of you that want to check it out on our website, that's where you'll find it, and that's what the name of this product will be under. So before I get started in the presentation, I just want to give a brief background onto why single-cell RNA-seq is useful and why people are starting to use it more. And for example, one main aspect of why it's useful is that conventional bulk RNA-seq methods only really provide an average expression across the sample type, whereas using single-cell RNA-seq, we can actually sequence the gene expression profiles of individual cells, and as a result, be able to find some newer or rare cell type populations that may exist in a heterogeneous tissue or sample that we otherwise might not have been able to discover by using bulk RNA-seq methods. While that's not only limited to that for the talk today, we're going to mainly focus on how using our product and using the CRISPR-Clean can really enhance the identification of cell types within heterogeneous sample. While single-cell RNA-seq has become a useful technique in recent times, it does not come without its own limitations. And one of the main limitations of single-cell RNA-seq is how the noise confounds the biological interpretation. So if we look here on the left, what we've shown here is how the poor capture efficiency on a per cell basis influences the data. As a result, we can see that in reality, using a droplet-based method per cell, you only capture about 10 to 20% at max of the actual transcripts within that cell. And as a result, if we transition to the right in the middle here, we can see that compared to bulk samples, the single cell data is sparse and contains a lot of zeros in the data, not being able to capture as many transcripts as you would otherwise in a bulk RNA-seq sample. Therefore, allowing to the idea here, if we look to the right, all the way on the right here, how this noise is quote-unquote pervasive, meaning that if we take two cell types that are supposed to be very similar between one another and we plot them together on an XY graph, the capture of those gene expressions should be very similar, but in reality what we see is that they're very different and we get what's called dropout events, some over-dispersion, and some outliers, and that the R-squared value between those two cells is very low. As a result, there is a need to try and reduce that noise and get as much biological interpretation that we can out of the single cell data. So here I would like to give a brief overview of the CRISPR clean workflow and how it can be used in the 10x workflow as well to essentially reduce noise in RNA-seq data. So what we can do is we can actually use CRISPR to digest unwanted sequences. If we look here on the left, anything denoted in purple is a sequence of disinterest, or a sequence that we don't want in our final library. We can use CRISPR to digest it. Consequently, we can do a size selection step to remove those short fragments and only PCR amplify the fragments of interest that we want in our final library. And what's great about this is that it fits very nicely within the 10x droplet workflow and only adds an additional 2 hours of time with the 45 minutes of hands-on time in the workflow. So before going into the presentation any further, I would like to give you guys an outline into exactly what's going to be covered in this presentation. And the first thing we're going to be going over is a proof of concept using cardiovascular samples. And essentially what we're aiming to do here is assess whether or not the sample heterogeneity can be captured using CRISPR clean technology, and therefore from there use in silico modeling using publicly available data, identify what are things that we can target using this technology to further enhance the reads being redistributed, and finally tie it in all together, assess the application using immune samples, and how we can further identify cell types. So what I would like to highlight here is how single-cell RNA-seq only utilizes the reads that align to the transcriptome. So what I mean by this is how if we look at the bar in green, we can see that these represent the total reads that come off the sequencer. So we have all of our reads, which represent 100% here. After we take those reads, we align them confidently to the genome, and we can see that approximately 90% of those reads align confidently to the genome on average. And from there, we take that genome alignment, and then we'll use a popular method such as Cell Ranger, which is standard in the 10x workflow. And as we can see, on average, approximately somewhere between 50 and 60% of the reads that align to the genome, therefore align to the transcriptome, and those reads are what is therefore being used for downstream processing. So in reality, you're only capturing about half the information off from the reads that you get off the sequencer for downstream processing. And so you might be asking yourself, well, how can we actually make this better or get more transcriptional information out of this? Well, what we notice is that even though we're only taking polyadenylated transcripts in the 10x workflow, inadvertently, some ribosomal RNA transcripts do get captured and mapped to the genome. Additionally, there are polyadenylated ribosomal and mitochondrial protein coding genes that get aligned to the transcriptome as well, but those genes are less informative and don't contribute to much in the downstream analysis. As a result, these make for easy targets for us to be able to deplete because they also make together approximately 36% of the total reads in the data. As a result, by depleting those reads from the libraries, we can therefore redistribute them to other informative transcripts and therefore boost our biological interpretation. Here, I would like to highlight how we can directly compare our depletion protocol with that of the standard 10x workflow. So at the top, we can see here we have our channels. So each coronary and pulmonary represents a different set of channels that was run through the 10x chip. From there, we take our amplified cDNA and we split that cDNA at the WTA step, and we either proceed with the standard 10x workflow or the depletion. Not explicitly highlighted here, but I want to stress that we're actually taking the same from the same channel and from the same WTA, splitting that between to proceed with the standard 10x workflow and that depletion to have a direct comparison of the same cell population and from the same cDNA. As a result, we then proceed either with the 10x workflow or our depletion, and with our depletion using our ribosomal RNA and ribosomal mitochondrial protein coding to eliminate those transcripts from our final library and therefore process it in further sequencing. What I would like to do now is focus your attention here to the bar graphs on the left. And what we have shown for you here is how the reads have been redistributed once coming off the sequencer. So out of 100% total reads in the top grouping or cohort that we have of each replicate of each coronary versus pulmonary cell type, we can see that after depletion, we see percent reads that are aligning to the transcriptome is approximately about 70% of reads. However, if we compare that to either a mock treated sample or a control, the control meaning a standard 10x workflow, we see that we're actually getting a redistribution of what we expect or hypothesize, which is about 50%. However, the key takeaway here is that with depletion, just in terms of informative transcript alignment, we get a boost to about 70% of reads confidently mapping to the transcriptome, which is great. As a result, at approximately 57,000 reads per cell depletion, we're able to recover over 200 more genes per cell, more UMIs per cell, as well as more genes that are contributing to biological signal, therefore allowing for more biological interpretation downstream. The first thing that I would like to highlight here from this figure is that again, we are working, and I would like to reiterate that we are working with endothelial samples. So going in, we are working with one major cell type. So as we can see, looking at these UMAP plots, and again, just to get everybody on the same page, a UMAP plot is just a two-dimensional representation of the differences and similarities of cells in transcriptional space. So as we can see, we only get one major cell type here, with many different cell types or subclusters or subcell types existing within that one major one. Now, the second point, and the other thing I would like to mention here or highlight, is the fact that how proceeding with depletion, we're not negatively impacting that interpretation. Again, so after comparing between the standard and depleted, we should see only one major cell type with many different subtypes existing within that cell population, but and as a result, depletion is not negatively impacting that result. Next, I would like to talk about how depletion is able to remove the technical noise that would be interpreted as a phenotypic signal. So what's interesting here is if I take the UMAP plots from the standard and depleted and do differential gene expression and compare, we see some different results. So if we focus your attention here to the left, we take a UMAP plot of the standard, and again, each color represents a different subcluster within the main cell type. And if I just pick one at random, pick this orange subcluster here, and do a differential gene expression, we can see that we have some very highly significant genes associated with the differential gene expression that are being up and down regulated in this case. However, if I compare that to the depleted and take that same subcell type and do the differential gene expression, we can see that those same genes are not as highly differentially expressed. So what does that mean? What that means is that we've essentially removed the technical noise and we've increased the capture of some of the lowly expressed transcripts, therefore reducing that technical noise. However, it's not affecting our biological interpretation in this case because we're still identifying it as a different subcluster, but we get a better, accurate, and more accurate representation of the differential gene expression between clusters as a result. Again, I would just like to highlight here and just reiterate the notion of how depletion is able to remove the technical noise that would be interpreted as a phenotypic signal. So if we look at the standard here and look at a different subcluster of cells and do the differential gene expression, we see a similar pattern where we see some very highly differentially expressed genes. Now, why might that be the case? Well, as we've shown before with the depletion, we get more UMIs per cell and we get more genes per cell. So the reason that these genes are being so highly differentially expressed is partly due to the fact that we're not capturing as many genes of the lowly expressed genes. However, with the depletion, we're capturing more of the lowly expressed genes, giving a more even distribution as a result, removing some of that technical noise and getting a better interpretation of the actually differentially expressed genes in that subtype population. Again, we're just removing the noise here as a result, but we're still keeping the biological interpretation that these are in fact different subtypes or subclusters of cells within the main cell type population. And so, if you remember before, these endothelial samples actually consist of both coronary and pulmonary endothelial cells. One of the main goals of this is we wanted to look at and capture, using this technology, the differences in the heterogeneity between these different cell types. Now, given they are both endothelial samples, we should see them overlap very nicely. Now, given that the coronary and pulmonary were run on different 10x channels, we have to implement a batch correction method in order to do the downstream analysis. So if you look here on the left, we can see that if we look at our standard and we compare the coronary versus the pulmonary after batch correction, we see some differences here. So what I've highlighted here and circled on the left is these two different types of populations that are distinguishing the coronary versus the pulmonary. So kind of more so directing to the fact that the batch correction here is less efficient in the standard when comparing the coronary and the pulmonary. However, when looking at the depleted, after redistributing those reads, we're actually able to get a better idea of what the transcriptional landscape is of these coronary and pulmonary cells of these endothelial samples. As a result, we see that after batch correction, we see a very nice overlap between the coronary and the pulmonary. But we do see one distinct specific phenotype that is unique to the coronary epithelial cells, further highlighting the fact that with depletion, we're able to better implement our batch correction methods here while still analyzing some heterogeneity in the samples for further analysis to look at the differences between coronary and pulmonary. Rather than just looking at the UMAP plots, another way that we can actually visualize the differences between the coronary and the pulmonary here and compare them between standard and depleted is to look at hierarchical clustering. So if we take the top 4 different upregulated genes and the top 4 downregulated genes in coronary versus pulmonary and do hierarchical clustering, compare them between each cluster, in the standard we can see that it's very noisy and hard to interpret exactly which cluster is being driven exclusively, the differences between the coronary and the pulmonary. However, if we take that and compare that same thing to the depleted and do the hierarchical clustering with the same set of genes, we can very nicely, very beautifully see how we can actually distinguish one major, through the hierarchical clustering, one major cell type that is actually driving the differences here when we want to analyze the heterogeneity and compare these endothelial samples and find what's different between coronary and pulmonary. So without depletion, we would have been unable to really make an effective interpretation of how these genes are being affected in the clustering as a result. And one thing we want to make sure is that what we're seeing with depletion is actually a relevant biological signal, right? So what we've shown here is how depletion is able to enhance that signal to noise and clarify a distinct phenotype subgroup of coronary epithelial cells associated with plaque. So, if we do a gene set enrichment analysis here, and we can see that based off the gene set enrichment analysis of that specific cluster, and look at those genes that are upregulated in that cluster, we can see that these genes are very highly associated with biological processes that have been shown in bulk RNA-seq samples that are involved in blood cell morphogenesis, for example, and extracellular structure organization, involved in these endothelial samples and contributing to coronary artery disease. So all that being said, just aids our interpretation that, again, the signal that we're seeing is in fact biologically relevant and aids to our understanding for further analysis. And so with all that, we were very nicely able to show how we can use this technology to effectively assess the heterogeneity between samples. However, we want to take that even further and see, can we actually increase the biologically relevant information by looking at redistributing additional reads rather than just looking at ribosomal and mitochondrial genes? And looking at this same plot again that we've shown before, we want to ask the main question, where else can we pull some non-informative reads from? So what we can see here is that if we take reads that align to the genome that are not used for analysis. So besides ribosomal RNA material that is still left in the library, what we find is that there are some reads that align to the genome that do not align to the transcriptome, therefore being captured in our libraries but are not being used for any downstream analysis. Now, this could just be due to the fact that these are stretches of poly-A's that are just being captured and not having any, but therefore not having any informative transcripts associated with them, so they're being filtered out. Nonetheless, they're still showing up in the data, and these are things that are not used for analysis that we can easily target and increase our reads being distributed to informative transcriptome. Additionally, some other things that we can target are what we would note as non-variable genes. So these are genes that are typically housekeeping genes that aren't used in any cell type identification, or genes that are not variable at all, meaning between cell types or between cell types, there's really no difference in the amount of transcripts detected, therefore not being used in any downstream analysis. So we can typically those cells are filtered out anyway, so we can target those non-variable genes and therefore boost reads being aligned to different regions of the transcriptome. And so with that methodology in mind of combining all of these guide sets together to be able to effectively redistribute reads, we've implemented an in silico depletion of 14 publicly available SRA10X datasets and tested this to see if it's a viable option. As a result, what we showed here is that if we combine our ribosomal RNA and our ribomino, and again, ribomino meaning our polyadenylated ribosomal and mitochondrial genes, if we combine that together, we can redistribute approximately 42% of reads, which is something that we did previously, which we showed previously. However, what we want to also do is implement these unaligned reads that are aligned to the genome but being filtered out, as well as these non-variable genes. And if we do an in silico depletion and apply these same principles or these to depletion, we can redistribute up to 50% of reads being aligned to the transcriptome, which in turn allows us to get more transcript information. And so now that we've done our in silico depletion, have our proof of principle that we can actually target these genes to redistribute reads, we want to showcase this in terms of how we can actually do this in the context of PBMCs to identify new cell types. So we're performing this in triplicates. So we have 3 different channels being run with PBMCs on the 10X chip. From there, we take our amplified cDNA at the WTA step. We take that and we split it equally between our control, which is therefore going to be proceeding with the standard 10X workflow, or our depletion. As a result, we're able to directly compare from the same channel the differences between the control and the depleted in triplicates as a result. And afterwards, we have obtained our final libraries and proceed with sequencing and downstream analysis. And next, what we want to focus on here is show how the depletion, after we proceed with the 10X workflow, the standard and the depleted, and we proceed with sequencing, how we're actually able to double the amount of informative transcriptional reads. So very beautifully, we can see if we look at the pie chart in blue on the control, we look at our informative transcriptional information, and we compare that with the depleted, we're actually doubling or having 100% increase in the reads mapped to the transcriptome with the CRISPR-clean depletion that's being aligned to informative transcript information, whereas non-informative transcript information would be your ribosomal and mitochondrial protein coding genes or your non-variable genes. And now, if you remember what we did with the endothelial samples, you can remember that we've sequenced to approximately about 57,000 reads per cell, which is a lot of reads per cell. So you might be asking yourself, well, it could just be that you sequence at such a high sequencing depth. However, with this saturation plot, we really want to highlight the point that across sequencing depths, the depletion is able to increase the number of UMIs per cell and approximately by almost 2 times, so 1.5 times as much, right? And even in this case, with the PBMC samples that we're using, at a sequencing depth of about approximately 25,000 reads per cell, we're still able to recover more genes per cell, more UMIs, and more genes contributing to biological signals, suggesting that as a researcher, depending on what your sequencing capabilities are, with depletion, you're able to get much more breadth of coverage as a result. And again, one of our main goals here is to highlight how we can identify different cell types using this technology. And so what we want to show here is that we're using a cell annotation tool for PBMCs that were done automatically from predefined legacy markers. So the reason we're showing this here is because we want to highlight the fact that we're not manually annotating these cell types by ourselves, right? The key here is that we're using markers from a published repository containing over 1 million PBMCs. And again, this algorithm takes into effect genes that are specifically up and down regulated, as well as the frequency of abundance. So again, just to highlight the fact that when we're identifying cell types here in these PBMCs, we're using an automatic tool in an unsupervised fashion so that we can eliminate as much human interference as possible for an unbiased result. And so sequencing approximately 12,000 PBMC cells, again at approximately 25,000 reads per cell, we can see that if we compare the UMAP plots between the control and the depleted, what I've highlighted here for you on the left in the control is a cell type population that is not being identified in the control, that is being identified in the depleted. But if we just focus on the control right now, we can see these two arms that are branching off of these CD14 positive monocytes, but are not being labeled by this automatic annotation tool as a different cell type. However, with depletion, by redistributing those reads, we are actually able to increase the Euclidean distance of these branches, and as a result, get a different interpretation as well as identify two additional cell types, one being a CD16 positive monocyte, as well as a rare population of CD1C positive dendritic cells. Again, just adding to the notion that we're able to identify more rare cell types as well as more cell types that we are not able to accurately identify in the control that we are able to capture in the depleted samples. And so what I'm highlighting here is that we're actually implementing another way of doing an unsupervised cell clustering method that's using deep learning. So before we use an automatic cell annotation tool, but here we're even taking even more human influence out of the equation and using a deep learning algorithm to automatically do the clustering to see if we can actually, in a completely unbiased way, see if we still see the same result and confirm whether or not we see additional cell types in the depleted versus the control. And again, the main takeaway here, so rather than using a UMAP plots, it's using t-SNEEZE plots. Again, doing the same goal, just taking transcriptional landscape and representing it in two-dimensional space for cell clustering. We can see that in the PBMCs, for the control, we're only identifying 9 clusters, whereas we're identifying an additional 2 clusters with the depletion. And what I've highlighted here is the areas that are being divided up into different clusters. So that arm of cell cluster 1 in the control is now being divided up into cell cluster 4 in the depleted, and cell cluster 6 in the control is being further divided up into 5 and 6 in the depleted. Just aiding to the fact that we're increasing the Euclidean distance of these clusters and accurately representing the different cell types that are existing within these populations. Here, this algorithm also has a way of assigning probabilities to these different cell types. And so what we want to show here is that it's assigning these cell types with relatively high probability, where lighter color, so yellow meaning a low probability, and the darker the purple, the higher probability that it is a cell type. So we can see if we look at that, specifically that one arm structure in the control, we can see that it's with a very low probability, a relatively low probability of being assigned that cluster. However, after redistributing the reads with depletion, we're actually able to assign it with a much higher probability that yes, this is indeed an additional cell type, adding to our confidence here that the result that we are seeing is in fact real. And finally, one last question we want to answer is can we sequence less but recover the same information using CRISPR clean digestion? Now, if you remember briefly, what we showed before is that with this CRISPR clean digestion with the protocol, we're actually able to double the amount of transcriptional reads at the same sequencing depth. But can we sequence half as much but still retain the same amount of information? And what's very nicely shown here is how we're able to detect the same number of genes and UMIs per cell with half the amount of reads per cell in the depleted sample. So if you take our undepleted sample, and if we say we're sequencing at a depth of approximately 27,000 reads per cell, And we take that and use our CRISPR clean depletion in half. So if we only deplete to half the sequencing depth of 13,500 reads per cell, what we can see is that the genes per cell is approximately the same. So in an undepleted sample at a read sequencing depth of 27,000 reads per cell, the amount of genes per cell you recover is 1,353, whereas with the CRISPR clean depleted, it's 1,340. So roughly about half the sequencing depth, and you retain about the same number of genes per cell and approximately the same number of UMIs per cell, suggesting that you can do a 2-for-1 sequencing as a result. So you can sequence twice as much and for the same price. Now, how does that actually affect our biological interpretation at sequencing half the depth? Now, while we're still retaining the same number of genes and UMIs per cell at half the depth, do we actually attain still biologically the same relevant information? And what we see here is very strikingly that half the reads, the depletion is still able to recover 2 additional cell types. So if we look at that the UMAP plots rather, that we showed previously, if we have highlighted here, this population of CD14 positive monocytes and those 2 arms coming off of it, those arms are now being identified as 2 new cell types, right? And that's because we've redistributed those reads and and as a result, when it does the cell clustering, it's able to get a better representation of those being different cell types by increasing the Euclidean distance between them. As a result, we are now identifying, again, a CD16 positive monocyte as well as a CD1C positive dendritic cell, rarer cell type population with the depleted sample at half the sequencing depth. In summary here, I would like to highlight how CRISPR clean single cell RNA boost kit is generally able to increase the number of genes and UMIs per cell, as well as increase our informative reads in PBMC samples, as well as identify additional cell types. Additionally, one great thing about this is that we're able to recover the same information at half the number of sequencing reads. So as a researcher, you're able to use your researching dollars more wisely and effectively do a 2-for-1 sequencing as a result. And finally, just to drive the point home here, in conclusion, our depletion is able to recover more information as well as increase the signal to noise and statistically enhance our biological interpretation for any downstream analysis that we have and have more confidence in our results as researchers. I would like to thank Jumpcode Genomics for giving me the opportunity to work on such a great project, as well as the Pandey lab for the endothelial samples he so kindly provided, as well as the Transcriptome Genomics Core for allowing us to use their sequencing technology and platforms. Now, I'll stick around for anybody that has any questions, and I look forward to hearing from each of you.
Thank you, Dante, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. Our first question is, does the CRISPR clean reaction use all the cDNA from my 10x prep?
Yeah, so first I would like to say thanks for having me and letting me present. But to answer that question, the answer to that is no, it does not use all the reaction from the cDNA from the 10x prep. So we've optimized the protocol to work with the 10x standard, where you take 1/4 or 1/4 of the existing library and then proceed with the depletion in that case. So yeah.
All right, thank you. Our next question, does CRISPR clean depletion introduce bias to my gene expression readout?
Mm-hmm. Yeah, so when we incorporate our depletion protocol into the library prep, we do it after the adapter ligation step, after the cDNA has been amplified. And by doing so, we can effectively remove a lot of the PCR duplicates that exist in the library as a result. So that's what we're depleting, to try and get more unique molecules and more unique genes in our libraries. So as a result, when we take that deduplicated library and we plot it in a control versus depleted, we get an R-squared value of approximately 0.99, so very significant. So by doing so, we don't introduce any bias into the as a result from depletion.
Great, thank you. Next question. Is this kit compatible with the 5' chromium assay, and what about Visium spatial?
Yeah, so we've actually done experiments, and we've actually optimized it as well to be able to work with those different platforms that 10x offers with the 5' chromium as well as the Visium spatial. So any libraries that you generate with either of those protocols, we have shown that depletion can work with it. And specifically, especially for the Visium, that's something we're really excited about and currently working more on right now to be able to have be able to push something more into the market. But yeah, as to the short answer, yes, we have. It is compatible, and we have shown that it can be depleted with the libraries generated from those protocols as well.
All right. And next question, where can I find detailed information on this kit?
Yeah, so if you would like to find, so we have all of our information on our website at jumpcodegenomics.com, and then also you can reach out to our sales team at sales@Jumpcode Genomics as well, if you would like to. And I believe in the presentation, there's also some links that you can look to for the reaching out to the sales team or for our website for where you can find more product information here.
Well, we are going to wrap up with that question. Thank you, Dante. Do you have any final comments for our audience?
No, I just look again. If anybody has any questions or concerns, you can email me at dante.d@jumpcodegenomics.com or email our sales team if you have any specific questions. But again, on our website, we have all of that publicly available data, and as well as all the data we've generated thus far. So if you really want to know more about our product, please make sure to go to our website and contact our sales team as well.
Well, thank you again, Dante, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, Jumpcode Genomics, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 21st, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.